invited by Carol, who I met at a course a couple of years ago, who's asked me to speak with you today about a topic that is very near and dear to all of our hearts, the beginning of human life. I like speaking on this topic. I'm a developmental biologist, molecular biologist. I study the development of the nervous system and regeneration of the spinal cord. But I also teach human embryology to the medical students at the University of Utah. And I'm often struck when I present this topic how both pro-life and pro-choice people believe that this is a question that cannot be addressed by science. That fundamentally it's a matter of personal belief, religious belief, taste, politics, and nothing could be farther from the truth. So I'm hoping today, in the next 50 minutes, to convince you all that this is a scientific question, and one that actually can be very conclusively resolved by science. So what are we going to cover today? I'm going to spend just a few minutes giving you a very brief overview of human development. Since you're all medical students, you've had human embryology. This will just be a reminder of the salient events. Then I'm going to talk a little bit about what are the scientific criteria that are used throughout the scientific enterprise to define when a new cell type has formed, either in the laboratory or in the course of normal embryonic development. I'd like to speak a little bit about the question of sperm-egg interaction, and can we use these criteria to define whether a new cell type is formed. Speak some about the differences between organisms and cells, and then ask the question, is an embryo an organism? And if so, when does organismal function commence? So these are pictures of early mouse development, but I can assure you humans look very, very similar to this. Shortly after fertilization, you have a single-celled embryo with nuclei from both mom and dad. Mom here shown in pink and dad with a little blue star. This one-celled embryo will undergo very rapid cleavage. So within the first 24 hours, it'll generate 2 cells. And by about 3 days, it has generated 8 to 12 cells and is now known as a morula stage embryo. By about 3 or 4 days, there's been a change in the properties of the cells. They undergo compaction, form tight junctions, and as a consequence, you can have unidirectional fluid transport into the inside of this ball of cells, and it'll blow up like a soccer ball. And this stage is now known as the blastocyst stage, and it's an important stage for a number of reasons, one of which is that it's the stage at which we get the first 2 cell types. So we start out with this zygote, and by the blastocyst stage, we have the rind or the white part of the soccer ball layer of cells known as the trophoblast. And those are cells that will largely contribute to the placenta and embryonic membranes. And we have a small group of cells on the inside, creatively called the inner cell mass. And these are the cells that are going to contribute most of the tissues that you carry around in the postnatal body. So almost all of the tissue that you currently have in your body came from this group of cells, the inner cell mass. Once implantation has occurred, development proceeds very, very rapidly. These are pictures of actual human embryos taken from the Carnegie collection, showing this extremely rapid progression of the development of form. So here, by about stage 18, which is around 45 days, you can already see the formation of eyes and limbs. And by the completion of the first 8 weeks of development, we've built all of the rudimentary structures in the body. So the primordial or the primitive stage of all of the tissues and organs exists. And that's by definition the end of the embryonic period of development. After that, we're going to have growth and maturation of those tissues during the next 2 trimesters. But that's fetogenesis, not embryogenesis. One of the things that this picture really illustrates is that thinking about when life begins, you immediately bump into this continuum problem. The fact that development proceeds so continuously from the 1-cell stage up through birth and, of course, beyond, leaves many people to more or less point to some arbitrary state and say, well, here, for me, that's developed enough for me to say it's now a human being. And that's where I'm going to say life begins. One common point is the formation of the blastocyst, because that's about the stage at which we get implantation. And many people are comfortable with implantation as the beginning of human life. If not the blastocyst, implantation itself, the establishment of a relationship with the mother. Many people like to point to the beginning of gastrulation or the formation of the primitive streak, because this is the stage at which the first actual restrictions in developmental fate within the cells that are going to give rise to the body occur. So we form those first three primitive germ layers, and many people will say, well, that's the very beginning of the formation of the brain. It's the beginning of the formation of the body, and that's when life begins. The brain is often pointed to as an important structure. I think this is because all of our most intimate experiences as humans, our consciousness, our thoughts, our memories, our emotions, all require the function of the brain. And because these are experiences that are so dear and near to our hearts and our identification of ourselves as individuals, maturation of brain or the onset of mature patterns of brain function are often identified as the point at which relevant human life begins. But you can go later than that. Peter Singer famously argued that you don't really have a human being until you have an individual who's capable of self-awareness. And based on some rather sketchy principles, he defined that as approximately 28 days after birth. Some people even go later than that. I'm a neuroscientist. Brain maturation, we know the brain continues to develop for the first 25 years of life, so several people in this room might not yet be human beings, as I constantly warn my teenagers. Send you back to the human parts factory. You're not a person yet. But the point of all of this is that choosing among these different definitions is really just a matter of utility and taste. It's not a matter of science or reason. And this is the central problem, or the central justification that people will give for saying the question cannot be resolved in anything other than an arbitrary manner. So how do scientists look at this question? In order to answer the question of when does human life begin, we have to ask a simpler question first, which is how do scientists decide when a new cell is formed? And I can tell you quite definitively that there are only two criteria. So all changes in cell type, whether they be due to specification, commitment, or differentiation, are detected by science either by changes in material composition, so what a cell is made out of, most typically changes in gene expression, or by changes in cell behavior, or by both. And these two criteria, changes in material composition and changes in behavior, are used throughout the scientific enterprise to determine when a new cell has been produced, either in the laboratory or in an embryo. So working in the nervous system, if I were going to hypothesize that a new cell type exists between neural crest and sensory neuron progenitors, I would have to define that new cell type by referring to one of these two properties. I would have to have a marker gene that's uniquely expressed in that population, that's turned on at the point in time when I say that cell type arises, or I would have to show a significant change in the behavior of the cells associated with this purported new state that they have entered into. So these simple criteria leave us with some important questions about when life begins, when human life begins. First, in order to get a new cell type, we have to get rid of the old ones. So when, in the interaction of sperm and egg, do the gametes cease to exist? And by the standard scientific criteria I've just defined, is a new cell type produced after sperm and egg cease to be? And lastly, what kind of a cell is it? So let's consider the gametes for a few minutes. This is a picture of a cumulus oocyte complex, so an ovulated, mature egg cell surrounded by follicular cells that are ovulated along with it. This ovulated human egg, with its associated granulosa or follicular cells, survives for about 24 hours, after which it simply dies. So that's the natural lifespan of a human egg. We spend 50 years making them, in my case, and once they're ovulated, they have a 24-hour lifespan. Similarly, these are human sperm, a scanning electron micrograph of some human sperm. And unejaculated sperm also survive less than 24 hours after they're fully mature. If you're fortunate enough to find yourself in a female reproductive tract, sperm will survive a little bit better. So they'll last between 3 to 7 days, depending on the stage of the menstrual cycle that the woman is at and other poorly defined factors. But overall, if fertilization does not occur, the gametes don't have a long projected lifespan. But if fertilization does occur, what happens? So this is a picture of a single sperm fusing to the surface of an egg cell. And upon sperm-egg binding, the most immediate and important thing that happens is that the plasma membranes of these two cells will fuse. This is a very rapid event, occurring usually in about 250 milliseconds. And when that fusion occurs, all of the protein and lipid that used to be present on the surface of the sperm, many of those molecules being unique to sperm, will be incorporated into the surface of this new cell. So there's an immediate change in the molecular composition of the cell surface. The sperm, of course, ruptures in this process, and it ejects all of its intracellular contents, including the nucleus, into the cytoplasm. So again, by this criteria, this new cell, we can call it a new cell precisely because there's been this very rapid change in the molecular composition of this cell. So the sperm clearly, as a separate cell, ceases to exist at the moment of sperm-egg fusion. And what about the oocyte? So following fusion and destruction of the sperm, the resulting cell has a new composition that's distinct from an egg. We've already mentioned the fact that the lipids and proteins that are characteristic of sperm are now incorporated into the membrane of this cell. But even beyond that, this cell has now a new nuclear composition. So it has two nuclear compartments, a blue one from dad and a pink one from mom, containing DNA that it didn't have before. So again, a change in the molecular composition of the cell. And this newly formed cell also radically changes its cell behavior and acts in a very un-egg-like manner. So the whole reason for an egg to exist is to attract a sperm and to fuse with it. But nearly instantaneously after the fusion of sperm and egg, within the first 1 to 3 minutes, the cortical reaction is initiated. So there's a change in calcium concentration within this cell, which induces fusion of membrane-bound vesicles near the surface of the cell to the surface. And those little bubbles of membrane will empty their contents into the extracellular space. And the contents of those vesicles are predominantly enzymes that will modify the molecular composition of the zona pellucida, this proteinaceous coat that surrounds the ovulated egg, and also modify the surface components of the egg itself so that no more sperm can bind to it. So antagonistic to the function of gametes, this new cell directly acts to prevent any additional sperm from binding to the surface. So I think we can conclude from this that the egg as a separate cell has ceased to exist, and that a new cell with unique material composition and unique behavior is formed at this very rapid instant of sperm-egg fusion. So what is this new thing? Part of the problem people have thinking about when does human life begin is you look at the immediate products of sperm-egg fusion and you say, well, that's just a cell or a ball of cells or an inflated soccer ball-ish kind of ball of cells, but it certainly doesn't look like a human being. So people will ask legitimately, is this new cell a new individual human being, or is it some other kind of cell? Maybe a pre-embryo. And it's motivated, this confusion about the nature of this cell that's formed by sperm-egg fusion is motivated because at early stages, recognizable human form and function are not evident. So how do we know that a zygote or a morula or a blastocyst is really a human being and not merely a human cell or a ball of human cells? This, of course, then brings us to the question of what is the difference between a full human being and merely a part of a human being, a human cell and a human individual. So I'd like to spend a little bit of time talking about things that don't distinguish embryos from cells. And there are at least 3 classes of things that seem very intuitive to many people as criteria for whether we have a human being or not, but actually are not good criteria. And these 3 properties are the ability of a cell to produce many different cell types, complex kinds of cell behaviors, and complex kinds of cell interactions. So we're going to go through each of these quite briefly. So producing many different cell types. This is a picture of a stem cell in culture. This little arrow is pointing to a barely distinct little blob, which when you blow it up is hardly more distinct, but this is what most cells look like in culture. And if you leave the cell alone, so it's been isolated from all of its neighbors, it will produce a clone of itself. So it will divide many times, and its progeny will divide, and eventually you'll get a pile of cells, but all of them will have exactly the same properties as the original cell. So this is how the vast majority of human, and in fact all animal cells, behave. If you isolate them, they produce copies of themselves, but that's all they ever do. However, there are some exceptions to this, most notably stem cells. So stem cells, or some classes of stem cells, have the property of pluripotency, which means they have the potency or power to produce many or pluriderivatives. And this is an example. So these are all cells that were derived from an initial isolated stem cell. It's able to produce cells that have neurofilament protein, so neuronal-like cells, produce cells that look like glial cells within the central nervous system, produce cells that have properties of smooth muscle and properties of adipocytes. So all of these derive from an individual cell, and yet I don't think anyone would argue that a cell that can do this is an embryo. These cells are produced in a random, chaotic manner. You can produce perhaps all of the cells in the body from certain kinds of stem cells, but you produce them in this disorganized mass that has far more similarity to a tumor than to an embryo. So producing many cell types is not a sufficient criteria to define the difference between a cell and an embryo. What about complex cell behaviors? Well, I'm going to show you this movie. I love this movie. It was made in the 1950s by David Rogers at Vanderbilt University, and it was converted from a 8mm film to a little video. What you're going to see here is a human cell in culture. There are actually 3 types of cells in this video. This cell here is a neutrophil. These anucleate cells are, I should test you, right? So class, what are these cells? These are red blood cells, of course, right? And then there are little tiny cells, bacterial cells. And what this video is going to show us is that you can remove cells from the body completely. And yet, built into those cells are certain properties that will give rise to complex kinds of behavior and complex interactions between cells that reflect the organization built into the cell in the body itself. So you're going to see this neutrophil in an isolated tissue culture dish, still acting exactly the way neutrophils like to act. So here he goes. So the purpose of neutrophils in the body is to search out bacterial invaders. This cell is exhibiting very complex behavior. It's navigating. It's obviously sensing chemical signals emitted by that bacteria. And it's chasing it, despite having to go around many, many obstacles. The suspense is killing me. Okay, he gets it. There you go. So this behavior, this complex interactive behavior, is simply the property of a cell. And seeing the complex cellular behaviors is, again, not a sufficient criteria to call this neutrophil a human being. So when cells are removed from the body, they continue to manifest their own intrinsic organization, i.e. they show the properties of cellular life, including complex adaptive behavior, coordination of their subcellular parts, and manifestation of characteristic form. Lastly, cell interactions. Cells present in tissues that are removed from the body continue to exhibit the same properties they had in the body as a whole. And consequently, interactions between cells within tissues can be quite complex, and they can, in fact, even generate new cell types. And complex functions such as growth, differentiation, morphogenesis, and wound repair can be seen in tissues based on the intrinsic properties of cells. This is a confusion that applies both to the beginning of life and also to the end of life. Because many of these kinds of arguments, well, you can't tell me someone's dead because their hair is still growing, or they're still healing wounds. These are very compelling arguments to people. And yet I hope to provide you in just a few examples evidence that these kinds of interactions cannot constitute a definition of who's a living human being and who isn't. So let's look at a few examples. This is a 3D reconstruction of a mouse embryo at about 10 days of development. And you can see the beginning of the formation of the fore and hind limbs. So these little buds or protrusions from the body are the earliest stage of leg development in mice and also in humans. We look very similar to this at this stage. You can take these limb buds off of the embryo and put it into culture. These were isolated about embryonic day 10 and a half. And at this stage, these limb buds are very, very simple structures. They contain only two types of cells, a very simple pseudostratified epithelial coating and an undifferentiated mesenchyme core. And yet, if you culture these cells for 7 days in the laboratory, this is what you get. You get a remarkably well-formed mouse limb that contains all of the characteristic bones, most of the muscles, connected appropriately. So this is a startling result in many ways. It tells us that that very primitive little limb bud with just two types of cells in it contained all the information it needed in isolation to go ahead and make this very complex structure containing multiple different parts and to organize those parts relative to each other so that you get a functional limb bud developing into an actual mouse leg. I'll give you another example. So limb formation is remarkably normal, both in terms of its morphology and also its patterns of gene expression. Similarly, if you take a piece of endoderm from the region that would normally form the trachea and put it into culture in the presence of an adequate inducing signal, it was actually discovered, you should note the date down here, 1967 was when this experiment was done. It was actually discovered by this group that many different kinds of mesodermal tissue, so long as they provide a cellular source of FGF signaling molecules, can induce lung formation in the laboratory. So tissue from the trachea forming region of endoderm combined with mesoderm forms anatomically and molecularly normal lung structures in the laboratory. And surprisingly, even the branching pattern and the biochemical maturation of this tissue, which are very specific, are replicated in culture. So as most of you will remember from human anatomy, lungs are asymmetric, right? And we have more lobes on the right side than on the left side. And as you can see, there's more branching in this little primitive lung produced in the laboratory on one side than on the other. So even the left-right asymmetry of lung formation is driven by the properties of the tissue itself. And this is just a cartoon of what normal human lung formation looks like at about the same stage. Last example, wound repair. If you excise skin from early embryonic stages, but even from adults, a lot of work has been done on wound repair because of the obvious relevance of this problem to both injury and also to warfare, actually. The military has done a lot of this research. You can injure it in the laboratory. So here you see a piece of skin that's simply been scratched to produce a wound in the middle. And if you let this piece of tissue go all by itself, it will replicate all the normal stages of wound healing, forming a very similar structure to the kind of scar formation that occurs in your body. So healing of wounds is not a criteria that we can use to distinguish between skin in the laboratory and a living being. So conclusions from these examples, I hope, are that cells and tissues preserve many normal properties and normal functions after they've been removed from the body. And these properties reflect the molecular composition of the cells themselves. So observing functions and interactions that can be explained based on the inherent properties of cells is insufficient to conclude to the presence of a human being. So where does this leave us? Do we have any way we can distinguish between human cells and human persons, human beings? I would say yes, and we can distinguish between them by factual observations about behavior. So how do embryos behave compared to how do cells behave? And the most relevant property of embryos is that they behave like this. So we start out with a single cell, and in an appropriately supportive environment, it will go forward to produce one of these. And it doesn't produce one of these in a random or chaotic manner, but
Dr.
it produces...
Condic, I'm sorry to interrupt. We have a question from Temple.
Yes.
Are we allowed to ask questions right now, or should we wait till the end?
It's up to you.
I think we can wait till the end, if that's okay.
Okay, that's fine. Thank you.
So in an appropriately supportive environment, the embryo undergoes a self-directed and organized pattern of development to generate the fetus. So it functions not as a single cell or even as a group of cells, but rather as an organism. So let's consider the properties of organisms a little bit more carefully here. So what is an organism? Well, Merriam-Webster's gives us a couple of definitions. First, a complex structure of interdependent and subordinate elements whose relations and properties are largely determined by their function in the whole. Or second, an individual, constituted to carry on the activities of life by means of organs separate in function but mutually dependent, a living being. So the features of organisms that distinguish them from cells are the following. First, they show growth towards a characteristic mature form. So ancient philosophers used to like to refer to this as a perfection of form. But what we know as scientists is that if you start out with a human embryo, it doesn't mature into an oak tree or into a baboon, or vice versa if we look at the embryos of those organisms. They have a mature characteristic form, and they develop towards that mature form from the very beginning. They show repair of injury. They show adaptation to changing environmental circumstances such that the overall function of the entity is preserved. They show coordinated function of all parts for the good of the whole. So all of these characteristics are related to that very definition I just read you. The definition emphasizes the interaction of parts in the context of a whole. And all of these things, growth towards a mature characteristic form, repair of injury, adaptation, and coordinated function, all require an organism to have some sense of identity as a whole. Now, using a word like identity makes a lot of people really nervous, but this is a matter of simple, factual observation. This isn't a matter of me imposing my definition on the embryo. It's a matter of me watching what the embryo does and naming it appropriately. So these functions require a wholeness to exist in this embryo that is resistant to perturbation. So we're gonna walk through each of these and ask, does an embryo actually meet these criteria? So when does organismal function begin? I think most of us would agree that probably this is an organism. And I think most of us would even agree that this is an organism. But where the real difficulty comes about is when we go back early enough in development that we don't recognize this thing as a human. So is this an organism? And I would like to argue that unambiguously from the one cell stage forward, embryos are clearly organisms. So why do I say that? Well, I say that because they meet all of these criteria. Growth towards a perfection of form is fairly obvious. We've already looked at the Carnegie stages of embryonic development. And this is, in fact, exactly what embryos do. They begin a process of development. And development, the entire business of development, is to produce a characteristic human form. They go about this unambiguously, directly, and right away from the very beginning. And they accomplish it within 8 weeks, which is pretty impressive. Repair of injury. Embryos are remarkably good at repairing injury, and I'll give you just one example. This is a picture of an embryo, a human embryo undergoing Ph.D. or prenatal genetic diagnosis. So what you're seeing here, actually I'll use my computer pointer, is a polished glass pipette here that's holding this embryo immobilized, while another sharper pipette is being inserted to remove a single cell from this roughly 8-cell stage embryo. So during PGD, a single cell is removed from this embryo. Now, you really have to think about this. This is like the most catastrophic injury you could imagine. I mean, imagine if a patient is brought into the emergency room missing an eighth of their body. Right, just missing. Chances are, no matter what part of the body you might imagine this to be, Survival probability quite low. And certainly not repair. I mean, if you lose something non-essential like your legs, you're not going to grow them back. But remarkably, these biopsied embryos survive, and if they're allowed to develop to the blastocyst stage and transfer to the uterus, they will regenerate these missing tissues. And there have been many thousands of live births from such biopsied embryos worldwide. So this is a pretty impressive ability to repair injury. Adaptation to changing environmental circumstances such that overall function of the entity is preserved. Again, I would argue that embryos are quite remarkable in their ability to sustain evolutionarily abnormal environments, including, for example, the entire business of in vitro fertilization. The very fact that we can make an embryo outside of the body is pretty impressive. But they can be even more impressive than that. I'll give you just a single example. So in 2003, ectopic pregnancies are usually don't survive, but occasionally they do. And this little girl here, Nahalanisi of Philippi, South Africa, was born in 2013 after developing entirely within her mother's liver. So as a little fertilized one-celled embryo, she escaped from the fallopian tube and was able to successfully establish a placental circulation with the liver. And she was delivered, I can't even call it by cesarean section. She was delivered by surgery, abdominal surgery, taken out of her mother's liver, and is a perfectly normal little baby. So that's pretty shocking. If you can produce a normal baby in an environment as atypical or unnatural as the liver, then I think this speaks very highly of the coordinated adaptation of embryos. The last feature I'd like to spend some time on is the coordinated function of all the parts for the good of the whole. So if we look at early embryonic development, what you see is that there's differences between the cells that are produced by the embryo from very early stages forward. So this is, again, a mouse embryo, but human embryos look very similar to this. They divide within the first day to produce 2 cells. And these authors have just pseudo-colored these cells. So embryonic cells don't generally come in pink and blue, but they're coloring them for you so you can follow the derivatives of these cells. In many embryos, what you see is that one of the cells gets a developmental advance on the other one. So it will divide earlier. The so-called leading blastomere will produce 2 cells before the lagging blastomere divides. And if you follow forward the progeny of these cells, there are differences in what they produce. So these leading cells contribute predominantly to or disproportionately to the inner cell mass, whereas the progeny of this lagging blastomere will contribute predominantly to the trophectoderm. So how does this come about? I mean, it's an observation, but what's the basis of it? Well, the observation is supported by a lot of molecular characterization of these early cells. So this was a paper looking at histone arginine methylation. So histones are DNA-associated proteins, and their methylation state or other post-translational modifications critically regulate their function in the chromatin. So either promoting or suppressing gene expression based on the modifications that you see. So what we're looking at here is a 4-cell stage. So here's a cartoon of the embryo. And these are just the nuclei stayed with Hurst, so you can see where they are. And then this is an antibody against histone H3 arginine 26 methylated form. And what you can see are 2 of these cells have a lot more staining, are a lot brighter, and 2 are a lot dimmer. This isn't just an artifact of the staining process, because if we look at a related histone molecule, histone H4, arginine methylated on arginine number 3, you see very similar levels of expression. If you quantify this, you find in red decreasing amounts of arginate, histone arginate methylation in different nuclei, and it's correlated very tightly with BRUDP incorporation, so with the global usage of the DNA in these cells. So not only do the cells have molecular differences at very early stages, at the 4-cell stage, but those molecular differences actually translate into different developmental behavior. They're using their DNA differently, they're producing different derivatives, And this is a coordinated process across all 4 of those cells. If you follow forward the consequences of these differences in gene expression at the 4-cell stage by the 8 to 10-cell stage, you find some other really important differences. So CDX2 and OCT3/4 are transcription factors, again, playing important developmental roles that are differentially localized by the 8 to 12-cell stage. So here in the late morula, you can see this green CDX2 staining is predominantly localized to cells on the outer rim of the embryo, whereas OCT3/4 is more broadly expressed throughout the embryo. And this segregation is known to depend on this difference in gene expression I described earlier at the 4-cell stage. So why is this important? Why do we care about CDX2 and OCT3/4? Well, we care about them because we know that those first 2 cell types, the cells that exist at the blastocyst stage and that enter into very different developmental pathways, the trophoblast making predominantly placental cells and the ICM making the cells of the mature body, those differences are critically dependent on exactly those 2 transcription factors. So we know that CDX2 will promote a broad pattern of gene activation that's characteristic of trophoblast. And it's also an autoregulatory gene. So the protein product of this gene will bind to its own promoter and enhance production of more CDX2. So it's a positive feedback loop. Once you have CDX2, it turns itself on and you get more of it. Similarly, OCT3/4 promotes a broad pattern of gene expression characteristic of ICM, and it also autoregulates. So once we have these genes turned on in the right cells, you can see how development will proceed in a coordinated manner. But how does this pattern arise? Well, it arises by differences at even earlier developmental stages. So if we go back in time, we find that CDX2 and OCT3/4 are expressed in most of the cells of the embryo. And in addition to their ability to promote their own expression, they are actual negative regulators of each other. So CDX2 will bind to OCT3/4's promoter and try to shut it off, and OCT3/4 will bind to CDX2 and try to shut it off. So there's sort of a tug of war. And they duke it out. It's believed that there's an apical signal, so something from the outside of the embryo, from that zona pellucida region, the surface of the embryo, that actually gives CDX2 the upper hand in those outer cells that are destined to become trophoblast. And once CDX2 has the upper hand, it can shut off OCT3/4 in those cells while allowing it to persist in the more medial cells of the embryo. And once the cells enter into these different pathways, they interact with each other to maintain a stable state of differentiation. So the cells that express CDX2 will inhibit OCT3/4 expressing cells from shifting allegiances and jumping over to the trophoblast lineage, and vice versa. So this is a really, really complicated set of interactions requiring multiple molecules, multiple cellular players, and spatial coordination of a pattern of gene expression that's critical to get this embryo heading in the direction of a mature human being. And based on this, I think we can conclude embryos are organisms. They are not just cells. They're able to mature to a characteristic human form, heal wounds, and adapt to novel environments. There are differences in developmental bias or lineage restrictions, certainly by the 2-cell stage. And by the 4-cell stage, there's evidence for globally coordinated molecular interactions and cell fate restrictions. So organismal function at the molecular and cellular levels. So I think this is very strong evidence. It does leave us with a little bit of wiggle room here, and that's the first 24 hours of development. So is there evidence for organismal function prior to this point, prior to the point of the 2-cell stage? So during the first cell cycle after a new cell has been formed by sperm and egg. So fertilization. This is a picture of an egg being attacked by many, many sperm. We need to ask the question, is fertilization a process or is it an event? And this is a medically relevant question because if you consult most medical textbooks, they would conclude that it's a process. And the reason they conclude that it's a process is because there are a number of unique events that occur only during the first day of life. So this unique period of development, which concludes with the fusion of the polynuclei and the first cell division, is its own little thing in the minds of many people. And a lot of people are comfortable calling all of those events within that first 24-hour period from sperm-egg fusion up to the first cell division, the process of fertilization. So we know that by about 20 hours, the two pronuclei, one from mom and one from dad, will move together in the middle of the cell. Their nuclear membranes will break down, and cell division will occur at about 25 to 33 hours, depending on temperature. And many people will call this the end of fertilization and the beginning of embryonic development. So is fertilization an event or a process? So at what point during the 33 hours between sperm-egg binding and the first cell division is a new organism formed? Well, many people are comfortable with this notion of syngamy, the end point of development of this fertilization period, as the end of fertilization and the beginning of life. So syngamy is the joining of the two pronuclei. It's the last unique event associated with the first cell cycle following sperm-egg binding. And I think it carries a lot of significance for people. First off, it has kind of this romantic significance. You know, we have the nucleus from the dad, the nucleus from the mom. They look at each other across a darkened room. They gradually move together towards the center of the cell, and then bingo, full union, fusion, generation of the zygotic nucleus. The whole process is done. So because it kind of relates to our sort of macro level romanticism, many people ascribe a lot of significance to syngamy. Plus, it's really cool. So let's just look at a movie of it. These are mouse eggs. And let's just focus on this one. Or you can focus on whichever one you like. You'll see the pronuclei appearing. Here you can see two. And they wiggle around a lot. They come closer and closer together. But then eventually, you see this really dramatic event where they disappear. And very shortly after that disappearance, the cell enters into the first round of cell division. So because you can see this event, it's very dramatic. Many people, and most medical textbooks, will define the formation of the zygote at syngamy. So the beginning of human life at syngamy. And many countries, for example, Germany, many states within the United States, Virginia, Michigan, define syngamy and the subsequent cell division as the onset of human life. But is syngamy really an event? And I would argue from a biological perspective, it isn't. First off, syngamy is just the breakdown of nuclear membranes. It's something that happens routinely at every cell division of every cell in your body. The only reason it appears to be significant in the first cell cycle is you have this nuclear compartments contributed by mom and dad. So it appears to be a joining, but it really isn't, because it's just the breakdown of the membranes surrounding the chromatin at that time. And it's necessary for appropriate mitotic spindle formation to occur, but it is not really an event. Moreover, you don't really have full union of the DNA at syngamy. So here in this experiment, the DNA contributed by mom and dad were colored different colors. So this cyan color is dad and this purple color is mom. And here you can see, prior to syngamy and after, we don't really have intermingling of this DNA, but the DNA is maintained in two separate domains. And these domains are maintained even at the 2-cell stage and even at the 4-cell stage. You don't have anything like, this is a normal somatic cell, the mixing of maternal and paternal DNA that you would see in the body as a whole. So the DNA from the two parents remains in separate domains and isn't really fused until at least the 4-cell stage, which leaves us with sort of a quandary. Are we stuck with this bad definition, or can we ask a better question? And I think we can ask a better question. So is the zygote an organism? So organisms are defined by global organization of parts for the sake of the whole. And the better question would be, does the cell formed by the union of sperm and egg exhibit global coordination of parts for the sake of the whole prior to syngamy? Or when does a pattern of behavior and gene expression initiate that serves the developmental goals of the embryo rather than merely the metabolic goals of a cell? So I want to remind you that cell membrane fusion is a very rapid event. These are illustrations of how snare proteins work for membrane fusion, and they're believed to work in at least a couple of different ways. These are the snares. They pull the membranes close together, and you can actually get a fusion of the lipid bilayer that creates a passageway. And as soon as this event happens, as soon as we open up a cytoplasmic channel between what was originally cell number 1 and cell number 2, you have a single cell with 2 different cytoplasmic domains, but because they're in communication with each other, this is a single cell. Or the snares can actually make a channel, a proteinaceous channel, that provides the same function. And this whole process, from attachment of the snares to opening up of a channel between the 2 cells, the fusion of those 2 cells, is an extremely rapid event, probably taking about 250 milliseconds. The molecular basis for sperm egg fusion is not currently known, believe it or not, but we know these proteins are present on both of the players, and so it's highly likely to be a snare-mediated fusion. So we get this event, 250 milliseconds from sperm egg binding to fusion, and then what happens? Well, a lot of coordinated events occur within this first 24-hour period. So within the first 1 to 3 minutes, we have this cortical reaction. So there's secretion or fusion of these cortical granules and secretion of proteins into the extracellular space. Factors secreted from the sperm are required for maternal nucleus to complete meiosis II. So as you recall from embryology, ovulated eggs haven't actually completed meiosis II. So they're arrested in that second meiotic reduction division. And we know that the sperm talks to the nucleus of the egg and induces it to complete meiosis. And within 1 to 3 minutes, there are changes in intracellular calcium that initiate the cortical reaction to prevent additional sperm from binding. So completion of meiosis and blocking of polyspermy are contrary to the functions of gametes, but they're absolutely required for an embryo to continue its development. Within the first 30 minutes, meiosis II is complete, establishing the definitive genome of the embryo, and sperm binding sites are destroyed. We also start modifying these nuclei. So the protamine that is present in the paternally derived nucleus is replaced by maternally derived histone. So we make a hybrid paternal nucleus that actually has paternal DNA, maternal protein. Maternally derived methylases will modify both of the nuclei to prepare them for transcription, but the paternal DNA is more rapidly and more extensively demethylated. So again, there's coordination between the nuclei before syngamy happens. The nuclei are talking to each other in terms of protein exchange, and they're also modifying each other's functions in a coordinated manner, because DNA modification is required for developmentally regulated gene expression that is only relevant to the functions of the embryo, not to gametes. Within the first 8 to 10 hours, we have DNA replication, converting both nuclei to a 2N state in anticipation of a cell division that's not going to happen for another 15 to 20 hours. And the onset of DNA replication is again dependent on factors from the sperm. So I'm emphasizing the role of sperm here because many times you'll hear that early development is driven entirely by maternal factors, which is why we call it a fertilized egg. And we don't refer to it as an embryo because the dad's contributions are really just irrelevant. And yet it's absolutely not true. Many of the early events within those first 24 hours critically require contributions from sperm. So it's definitely a coordinated function between maternally and paternally derived components. Earliest expression of the zygotic genome occurs immediately after DNA replication. And interestingly, the paternally derived nucleus suppresses expression of maternal gene. So transcription of paternally derived DNA is 4 to 5-fold higher than maternal. So DNA replication, differential transcription are required for the next step of development.